because some reason Zoom, if I actually make PDF full screen, then some reason I cannot change the pages. I don't know why. So I gonna present talk as this way. So just FYI.
Okay. Do you mind if before the talk starts, we show interval slides and go back to your screen at 5?
What do you mean? I'm sorry, I didn't understand.
It's okay. No
okay,
worries.
sorry. But can you see my screen, my PDF file? Is it okay?
Yeah, I can see it.
Yes,
Great.
looks good. Great audio too.
Great. Perfect.
Okay, so today we are fortunate to have Dr. Ruriko Yoshida, who is an associate professor of operations research at the Naval Postgraduate School. Her research topics cover a wide variety of areas, applications of algebraic combinatoric statistical problems, such as goodness of fit tests, optimized camera placement in sensor networks, phylogenetics and phylogenomics. Today, she's going to tell us about tropical principal component analysis and its application to phylogenetics. So Ruriko, the stage is yours.
Thank you so much for nice introduction. So thank you so much for this opportunity to give a talk today. Yeah, so this morning I changed like my mind, and it's a different topic than what I thought. This is a really exciting area for phylogenetics, phylogenomics, and that I'm very much interested in. So here we go. And please let me know if you have any questions. Oops. All right. Okay, I cannot go. All right. Okay, so what is phylogenetic tree? So phylogenetic tree is a tree representation of representing a evolutionary history of species or organisms. So for example, so this is actually kind of controversial data set, and we're gonna analyze that set of data set in the end. So you'll see this some examples, but so I'm pretty sure in high school, you are in high school biology classroom, you hear that our ancestor is coming from oceans, right? We used to be living, like our ancestors used to live in the ocean, and for some time of the history, they came to land, all right? So biologists are really interested in how this has happened. So one of the specific questions, this is a really controversial data set, and so this tetrapod, so you see this, and you see mammals and some like, reptiles, and so they are land living things. And so lungfish, this is a fish that having lungs, so they can actually kind of go on like land. And also this is the coelacanth. That's that, like you see, you might hear those fishes, they are living in the deep ocean. And so question is that like lungfish or coelacanth, which one of them is close to tetrapod? Okay, so that's a 2013 scientists generated whole genome of the African coelacanth, and that's actually published in the Nature. And immediately after that, people actually, sequenced that like genes and like compared with those species. And that's also published in the biology journal. That data set, we applied our method to that data set. And you'll see this actually at the end. Okay, so why it's controversial? Because like phylogenetic trees, so this is kind of phylogenetic trees, and we don't know. So there is no internal branch. So this is actually, so it looks like the picture looks like lungfish is closer to like tetrapod, but actually we don't know which one. So that's the like classic question. Okay, so this is one example. So phylogenetic tree reconstruction. So if you have one tree, or like, in order to reconstruct one phylogenetic tree, what we're gonna do? All right, so suppose we have input is that like a sequence of DNA sequences or RNA sequences or protein sequences, whatever. And what's going on here? From there, we actually like construct a phylogenetic tree. There are tons of methodology out there, like using continuous time Markov chain models. That's the parametric method, or some distance-based method, and there are tons of it. So in this talk, I don't talk about phylogenetic tree reconstruction method. So I assume that because since 1960, scientists were developing so many methodology out there. So I assume in this talk, we have pretty good phylogenetic tree reconstruction. Okay. All right. So this is kind of mathematical or like a statistical model for the like genome, how the genes are actually like generated. So suppose we have species trees, that's the relationship between species, how they are actually evolved and so on. And under that, we have the gene tree. So gene tree means that tree is, reconstructed. That's a tree, phylogenetic tree representing based on a particular gene. And also from there, we have the sequences. So if you are in the nature, we know the species trees. And from there, you can generate gene trees. And then from there, you generate the like sequences, DNA sequences or protein sequences or whatever. But in reality, actually, we only see the sequences. We don't know the gene trees and we don't know the species trees. So our real job, in the real life, we try to infer the gene tree, each gene tree from the gene sequences. And from all bunch of gene trees, we try to infer the species tree. That's kind of hierarchy of the statistical method we actually have. So in this talk, I assume that we have good estimate of the gene trees. Okay, we are not talking about gene tree reconstruction here. I assume that we have so good gene trees. So we have a sample of gene trees. Okay, so that's our problem. So now this actually motivates the like next step. That's called phytogenomics. Okay, so phytogenomics. That's that we are trying to do some statistical analysis on a sample of phylogenetic trees. So it's not one tree. I'm not doing this like, okay, I care. I shouldn't say I don't care. I care a lot for reconstructing one phylogenetic tree. But so many people are already doing this. So I actually care about developing some statistical method, statistical learning method on a sample of phylogenetic trees. So that's what we have. Okay. But the problem is that you notice trees are, trees. That's not the vectors. That's we're gonna vectorize. So I'm gonna let you know how we're gonna vectorize. So from like, okay, sequence to trees and tree to our process, we're gonna somehow vectorize. However, still how we vectorize the tree, we have to capture the information of the tree topology. Tree topology means the shape of the tree. So this is extra like difficulty for developing machine learning models. Okay, so this is the big question. So my question is developing machine learning models on a set of phylogenetic trees. Okay, I hope it's clear. So if you have questions, just let me know. Okay, so that's a question. All right, so again, language spatial data. So this is actually, I will show you the result at the end. So we have 1,290 gene trees, so about 1,300 trees, and number of leaves is 10. Okay, that's actually kind of simplified picture. But so 10 leaves, and we have the like about 1,300 trees. We want to actually do some statistical analysis, but like in the original paper, this data set is coming from, author did quick some kind of like statistical analysis. So like they just compare, check that like tree shape, tree topology, how they look like. So, but the bad news was that, all right, So we have a tetrapod, just group them together. And so like about 1/4 of the gene trees looks like this tree topologies, and 1/4 of them looks like this, and 1/4 of them looks like this, and 1/4 of them looks like this. So kind of hopeless, right? Because it's, you know, they can do, but this is like kind of, they look at like only, you know, tree shape, and just like they kind of counted how many of them, but it's not that informative. Question is that we want to know, this is a lungfish, by the way, lungfish or coelacanth, which one, sorry, this is shark, I'm sorry, this is a shark, this is coelacanth, and this is lungfish, and lungfish or coelacanth, which one is close to tetrapod? Okay, so that's what we want to know. All right, so this is kind of difficult question. So just we cannot just check the how what shape looks like, because phylogenetic trees, actually I'm gonna go back to a little bit. So you each actually branch has weight. So this is a weighted tree, and also internal node, they don't have label. So the weight kind of representing like mutation rates and also evolutionary history. So go back to this guy. So the author of this data set, you know, they just check that how tree shape looks like, but they didn't consider that like branch length of each branch in each phylogenetic tree. So how actually we're gonna do that? Okay, so that's the biggest question. So, you know, this is really motivated data set. And so there is a PCA. I'm pretty sure you guys know this, like principal component analysis, just a classic principal component analysis. That's basically developed by like Pearson, right? That's like early 1900s. Okay. So we just apply the classic PCA and just how it looks like. We just did that. And we saw that just this is it's kind of hard to see this. This is the 3-dimension. So we did the dimensionality reduction because that's a high dimensional data. And just like, okay, that's a 3-dimensional, you know, yeah, picture, it's 1, 2, 3, and you cannot really see this, but actually I saw this two, you know, separated like clusters. So I said, oh, that's great. This is why, maybe this is why, you know, we have this data set. Many biologists have like different, like, you know, conclusion, because some people say lungfish is closer to tetrapod, and some people say that coelacanth is closer to tetrapod. This is why. But I was actually looking at this. Wait a moment. That looks like shape of the tree space. And tree space is the space of all phylogenetic trees with fixed labels of leaves. So I was thinking, because, you know, I work on the phylogenetic tree, so I kind of knew the shape of the tree space. So I thought, that looks like kind of strange. So this actually picture motivated me to develop a principal component analysis over a space of all phylogenetic trees. So that's the really motivated picture. Okay. So I'm gonna go back to a little bit basics. So I'm pretty sure you guys know this, but I'm gonna review the classical principal component analysis. So principal component analysis is a great tool, right? That's so great tool to reducing the dimensionality, because so that we can actually see that how the data points are distributed over the space. Okay, so this is just picture. And so that's using, so that's a PC1 and PC2 over the three-dimensional things. And basically that, like we can actually reduce the dimension from three-dimensional data points to two-dimensional. Okay. So here again, this is PCA, classical PCA assumes Euclidean space. Okay, because this is supposed to be geometry, so I'm gonna talk about the geometry. Okay, so this is PCA, classical PCA assumes Euclidean space. It's very nice, so that like we can calculate eigenvalue, eigenvector, and it's so easy to calculate. It's really fast. However, we are talking about not Euclidean space and not just a straightforward vector, but I want to apply the PCA to a sample of trees. So like this. Okay. So that's extra challenge. Like I said, a space of all phylogenetic tree is a mess. It's hard. Believe me, I'll explain in a second. All right. So, but in order to explain the PCA, I have to tell you, I have to review something quick because this is a basic idea how we developed it. So the PCA is kind of similar to like regulation, but a slightly different way. So how are we gonna do this? So basically, like those blue points, they are data points. That's an observation. And the red points are also going to projection onto some lower dimensional plane. Okay, that's a Euclidean step because it's easy to explain. And so you see that this bar, that's the residual, that's the difference between blue dot and red dot. Okay. And PCA is actually, we can write down as an optimization problem. So the minimum, try to find lower dimensional plane such that minimizing the sum of distance between blue point and red point. So that's how you calculate principal components. Okay. All right. So now I'm gonna talk about a little bit tree space, and also in order to talk about like those statistical methods, I have to talk about the metrics, and I'll talk about that. So here in this talk, I assume that equidistant tree. What's an equidistant tree? So that's a rooted tree. So the distance from root to leaf, each leaf is the same. So for example, this is a root, and leaf A, that's a 2, and root to leaf B, that's 1 plus 1, that's equal to 2, and root to C, that's 1 plus 0.2 plus 0.8 equal to 2. So like that, okay? And we assume, this actually talk, I assume that all gene trees are equidistant trees. So is it kind of really over, like assuming like crazy? No, actually it's not, because think about evolutionary time. So each gene trees, like, ancestor to the, like, current species. So if we have the common ancestors, most recent common ancestors of all species you see in the present, they have the same evolutionary time, right? So it's not a crazy assumption. So this is I assume. And also it's called coalescent model. Coalescent model is a really classical model to model for those gene trees and species trees. So it's, and also the coalescent assumes the equidistant tree. So that's actually not a crazy assumption. So I assume that. All right, so how are we going to vectorize? So calculate the first this similarity map, that's the pairwise distances between leaves. So for example, like I have this guy. So that's the AB. So that's the distance from leaf A to leaf B. So 2 plus 1 plus 1, that's equal to 4. And I pick this guy, that's a BC, and that's a distance from B to C. So 1 plus 0.2 plus 0.8, so that's equal to 2. Okay, so you calculate that. And this is the N by N matrix. N is the number of leaves, in this case 5 by 5, and diagonal equal to 0 because each leaf to itself distance equal to 0. And also that's a symmetric, obviously, because distance from A to B is just equal to distance from B to A. So this is a symmetric matrix and diagonals are 0. So we just take the upper triangular part and we order the lexicographically. And this is the vector we consider. And actually, like a while ago, like long time ago, someone, Buneman, actually proved that if actually tree, like equidistance tree is there, then this is actually this vector is a ultra matrix. And also that if you see this vector, if there is a equidistance tree, then there is a one-to-one correspondence. So this vector is a unique representation of this particular tree. Okay, that's the basically one-to-one mapping. Okay. All right. So that now, our question is that we vectorize all trees and that's a one-to-one mapping. So that I'm gonna actually consider a space of ultra matrix. So that's the vectors like this. Okay. So that's that I call tree space for this talk. Okay. Then, yeah, so this is the ultra matrix. And so basically that this is the ultra matrix has some particular properties, and that is that this is a matrix. So that means that, there are some property to satisfy, including the triangular inequalities, and also that actually, this is a little bit stronger than properties than triangular inequalities. But also that like a bunch of algebraic geometers shows that this actually space, so I say space of ultra matrix, that's a tree space I call, this is a tropical linear space. That's a bunch of people proved this before. So now here I'm gonna talk about tropical. What does tropical mean? I'll talk about that. So this is why we are using tropical geometry to for the principal component analysis, because it's such a natural way to look at the tree space. Okay. All right. All right, so what's a tropical algebra or tropical arithmetics? I'll talk about that. So here I'm gonna use it so-called max plus algebra. So that is that. So suppose A and B's are real numbers, and addition. So this is actually ring. That's actually like algebraic rings, and I define the addition as a max and multiplication that's equal to plus. That's why we call max plus algebra. Okay, and then this is the distance measure or tropical metric that we call tropical metric between two phylogenetic trees. So we vectorize that and then like vectorize all equidistant trees, and then we have to define that distance between two trees, right? So here it is. Okay, so this is so we have the B and W. Those are ultrametric, so that's the vectors for representing, one tree and representing other trees, and then that's the basically we take max of this guy. And actually, if I can draw, yeah, no, I cannot really draw, huh? Yeah, so easiest way, I try to draw something, but I have to erase, so maybe I shouldn't do that, but we'll see how it goes. Oops. All right, so we can also show that this is equal to the max. It's much easier way to do it is the max of B minus W plus, minus, sorry, minimum of B minus W. If I do this, this is basically equivalent. It's much easier to understand. And I like this way. So, all right. Okay. So the also that I'm gonna actually consider that this is I'm gonna mod by just 1, 1, 1, 1, 1 vectors, because if I don't do this, this metric is not a metric. In order to make that this distance measure as a metric, I have to do this mod by 1, 1, 1, 1, 1, 1. All right. Okay, so here is an example. So suppose I have B equal to 0 and W equal to this guy, then okay, I should maybe do that. Yeah, I can do that. So I say that like, B minus W, max is 0, right? The max is 0. And because I have B minus W, that's 0, minus 3 and 1. And the max is 0, minus 3, that's a minimum. So this is equal to 3. Okay. All right, so that's how you calculate. And why I have to do this like mod by 1, 1, 1, 1, 1, because so suppose I have that like some vector, let's say like I don't know, x, that's equal to like say the 1, 1, 1, 1, 1. And I take the distance between the tropical, say that like x minus x and b. And you can kind of see this, that this is equal to 0. Right? And if x and b's are like different vector, that is not a metric, because in order to be metric, distance equal to 0 if only if they are it is the same vector. And so that's why we define that this v equal to x, or actually this v equal to any, that's also over this like vector space, that's gonna be CCC. Okay, that's any constant. So that's I meant was mod by 1, 1, 1, 1, 1. All right, I hope it's clear. All right, so that's the basic. Okay. All right. So we define one paper that like there are two types of tropical PCA. One is that like tropical linear space, and another one is tropical polytope. And here we talk about tropical polytope because it makes sense. And but we are actually developing the tropical PCA as a tropical linear space for the phylogenetic trees, or like space over the space of phylogenetic trees. I'm developing right now, but I'm gonna talk about the polytope version. All right. So what's a tropical polytope? Tropical polytope is basically like suppose we have finitely many vectors, and we define the tropical convex hull that's equal to this guy. So AI and AIs are real numbers and BIs are some vector in a set, and those are finitely many. And so I say like why we use this guy, because in a sense that like if you are talking about like some linear plane. Okay. Just a moment. I think I'm gonna do this. I'm gonna draw a bunch of stuff. But so typically, if you have that, B1 plus that's Euclidean space, by the way. B1, that's equal to some constant, like something like B. So that defines that like linear plane, right, from the calculus. That's for the Euclidean space. And if we, PCA can be represented like PCA plane or, like something like, some linear plane, linear space. So basically this is equal to 0, but that's generated by the principal component, can be written as this guy. But if I actually take the all like tropicalize, basically that becomes the tropical polytope. So in a sense, kind of makes sense to apply the tropical polytope as a tropical principal component. All right, so that's what we do. And also, we define the tropical geodesic as a tropical line segment. That's the basically tropical polytope generated by just 2 points, 2 vectors. Okay, so that's we say that. And if I do this, basically, like if we look at the space of phylogenetic trees, then this tropical line segment is intrinsic. That means it never comes outside of the outside of the space of phylogenetic trees. And also that if those vectors, all vectors in a V, if they are ultra matrix, or i.e. that's a vectorized phylogenetic trees, then this polytope is gonna be inside of the tree space. All right. I think I'm gonna talk about tree space in a second, but I'm gonna tell you little bit fact. All right. Tree space is such a difficult space. So basically, so this is a piece of paper, and you can imagine. So that's we have the Euclidean space, and tree space is that. So you are doing that like basically like origamis holding and lower dimensional polyhedral cones, basically union of the polyhedral cones, and it's not even convex, and it's not even manifold. It looks like this, basically. Just a bunch of those, just glued together. It's very complicated space. This is why it is so nice to have this property. That's why I'm using this, because, I want to do statistical analysis on a much lower dimensional, like polyhedral cones. I want to do some statistical. All data points are in this paper, not here. If some points are here, then that's not a tree. All points on these papers are trees, but here, not here. So I want to do statistical analysis on this like crazy like origami holding. That's I want to do. Okay, so this is a hard problem. This is why it's hard. But if I use that tropical polytope, then naturally that's gonna be inside. All like points inside of the tropical polytope, they are inside of this paper. Okay. All right, so this is just example. And oh, here is a picture of the tropical polytope. That's like, looks like classical polytope, but yeah, so this is the vertices. So rows of the matrix, that's the vertices of the, that's a tropical triangle. And that's the tropical example of the tropical triangle. And you will see this a lot of tropical triangle in a second. All right. So we have also formula for the projection, because remember, I want to now tropical PCA, I want to calculate the tropical polytope. This case, because I want to visualize, I want to find the tropical triangle. So the blue point distance, sum of the distance between blue point and the red point is minimized. So that's I want to find. Okay, that's my goal. This is my goal. Right, so this is I want to find. All right, so that means that I have to have the like projection, and there is a formula. So I'm not talking about the details. Just believe me, there is a formula. And the nice thing is that if I have the tropical triangle, then dimension, this is also important. It's really important point. You might think, of course, triangle has the dimension of, you know, just like one dimension, right? Or if I have a convex hollow of the S many points, dimension must be at the most like S minus 1. But, you know, you have to be careful. Some spaces, that's not obvious. But this actually tropical matrix, it is. And tropical polytope, it is. So this is a very nice property to have. So that's that also nice property. So that's why we decided to use that. All right. So also the tropical PCA, I can write as a mixed integer. Actually, I think, yeah, actually mixed integer linear programming program. So this is actually nice. Okay, so let's show you the main dish for that, like our paper. So, oh yeah, we also wrote some code we can write down. So I'm gonna skip this one. All right. So we actually you can download from my website, or I think we might have GitHub. But anyway, so if you want to have that R, we implemented everything in R. We can actually access to that, like our code. All right. So as I said, that this Langfish coelacanth data set, I'm gonna apply. We applied. Remember, in the beginning of the talk, we had the big two like clusters. I want to know that that's artificial or not. Okay. So also we develop another PCA, actually, my colleague and I developed with different metric. That's not a tropical metric, that's a BHB metric. And actually, this is also for the PCA over the tree space. You see this all points are here. But for that, bad thing about this metric is that seems like all data points are kind of in the corner. That somehow artificially just like clusters in a corner. So this is just one thing. So we have that. And also this is the tree topologies. So you see this, that's actually tree topologies of the all projected points. Not, so remember, PCA can be used for basically eliminating noise too. So that's kind of also we applied. So the tree topologies on the all projected points of the triangle using the BHB metric. This is the tree topology. And basically it's saying is that Langfish is closer to tetrapod. And actually recent papers show that that seems like agreement now. All right, so this is the tropical triangle. It doesn't look like triangle, but it is a triangle, tropical triangle. And it's good news for this metric. So I wanted to show you two different PCA over the tree space with different metrics. And one has that kind of clusters in a corner, but in this case seems like nicely distributed. So using those two different approaches of the PCA over the tree space, it seems like we saw that two big clusters using the classical PCA. That seems like artifact of the structure of the tree space because it looks like origami folding like this. So basically one distribution, one actually points are here and maybe one points were here. And since the Euclidean space, that's a three-dimensional space like this, much bigger space than tree space. So I think just it looks like big cluster, different cluster, but actually it is just one cluster. So this conclusion is that seems like just one cluster. All right, and I'm gonna skip this. And okay, so this is another good data set. This is called apicomplexer. I love this data set. So you might hear something like toxoplasma. You might hear this. Toxoplasma, I love toxoplasma too. They are parasites. I love parasites. They are awesome, actually. They are so great. So toxoplasma is that usually, they are in when they are actually kind of maturing, growing up, they are inside of the mouse. And when they wanna mate, they are actually controlling little mouses brain, and little mouse is getting attracted to cat's urine. So then, they're gonna be eaten by cats, and toxoplasma goes to the cat's, stomach, and they find a mate. And then those eggs or something, they are actually going to the cat's poo, and mousey, little mousey, they're gonna eat that cat's poo and come back to the mouse's body, and life goes on. I love toxoplasma. That's just fascinating species. They are great. So they are parasites. All right. And the apicomplexer, that's malaria too. Malaria is in this family. So this is a very interesting family of the organism. And just a moment. Okay. So here is the BHB matrix. That's one PCA. So you see this, still kind of, it's better than lungfish, but it's still kind of gathering. And here is our tropical triangle, and it's here it is. And again, actually, this data set is coming from my friend. So I talked to that my friend about the result, and they really like the result because so these three topologies are actually most common one on a projected tropical triangle, and actually this is the actually non-species tree for the apicomplexer. So this is a good result. And also lungfish too. Actually, I forgot to say that lungfish, the most common tree topology for the projected trees on a tropical triangle, actually the lungfish is closer to the tetrapod. So that's again consistent with the all other publications. So that's a good thing. Okay. All right, and also we actually applied to the influenza data set. This is a big data set. It's really gigantic. About, I think, 20,000 trees for each year. So we have 1993 to 2017. So we collected influenza flu data collected in a state of New York. Okay, so that's what we did. And so we got a fairly good. So this is actually compare against the BHB and PCA. And this is, we calculated the variance of explain. That means that the higher number, those numbers are between 0 and 1, and the closer to the 1, the better. Okay, so it is like super close. So this is fairly good. I mean, BHB really we didn't do so well. Sometimes we did really good, but they didn't do well. But actually, yeah, most cases like tropical PCA did actually. I think all of them, we have the better performance. All right, and here's some references. And yeah, if you are interested in, that's the references. And also, thank you so much for your attention.
Okay, very nice. Perhaps we can thank the speaker.
Thank you.
Okay, there are a few questions to ask. One of them is, can this work be generalized to a bigger class of graphs than phylogenetic trees? So does your method specifically use the fact it's a tree and not just a graph?
Yeah, actually, like this idea can be applied to graphs, I think perhaps, but I haven't applied, but theoretically, actually, we can. Some of the graphs, sure. And actually for the BHB, space, that also can be actually like several people actually apply to the graph. And yes, we can.
Great, okay. And a related question. So you were working on the space of trees, and moduli spaces and the like show up all the time in geometry. So can you do this kind of PCA on a general moduli space? What kind of structure do you need to be able to do this?
Can you repeat again? Sorry, I didn't understand.
So can you generalize this approach to the moduli space of things other than trees? It's sort of closely related to the previous question.
Yeah. I don't know. That's actually, I don't know. That's actually a good question, but I don't know. So actually here, I did that. We take the 1, 1, 1, 1 more modularized because just I had to do this technical thing, just a little bit technical thing. But we might be able to trick out because, all right, this is geometry question. So our answer, so little bit maybe tricky, but where was it? Yeah, so this space. So this space is actually isometric to R2 E minus 1. This is exactly isometric to. So maybe I can tease out a little bit, and we might be able to kind of, do something, but I don't know. I haven't tried.
Okay, great. Another question is, does this procedure work if you don't fix the number of leaves of your phylogenetic tree?
Okay, so tree space is that's a great question. This is actually like really, really problem too. That's actually we work a lot. So by definition, so BHB, I talked about the BHB. That's the Borella, Holmes, and Bogman space. And they developed this space like around, I think, 2000. All right, so that's like 20 years ago. But they defined the space of phylogenetic trees with fixed leaf labels. So now, this is actually a great question. I try to actually do something about that because it's really typical question that some of the leaves might be missing, and some of the leaves like, like extra for some particular genes. So in a real data set, they are much more messy. So what I did is that I take the bigger set of the like leaves, like, just I take the all possible label for the leaves I can observe from the sample, and then I impute like phylogenetic tree. So I wrote some paper about this. So we can do that this way, just by imputation. I can do that. That's the actually best thing I can think of right now. But this is a great question, and yeah, this is in a real program, in a practice, and that's just answer is that I do imputations for that. I take the bigger set, just all like I take the leaf set as all possible leaf labels from the observations, and then each trees I try to impute. But the problem is that that might actually, like computational power might suffer, and sometimes the superset of the leaves that might be gigantic. So I don't know that's the best way, but that's kind of goofy way, but we try to accommodate. But that's a real problem in the biology.
I see. Another question is, can tropical distance be used to define an algebra over nominal values?
Nominal values. Actually, quick answer is no. That's actually for the numerical values. But actually for the nominal values, I use that, like in the phylogenetics, like we use like maybe Hamming distances or some other distances. So I haven't really seen this tropical distances or tropical metric on the nominal value. No, I haven't seen this.
Okay, and I think the last question is, in hyperbolic space, the volume increases exponentially, meaning that trees embed well there. Is there any kind of comparison you can draw between this algebraic approach and the hyperbolic embedding approach?
Actually, like I've seen, this is not exactly the space, but so like for the tree reconstruction, so I talked about phylogenetic tree reconstructions, and so in actually tree reconstruction method, when we use that, like that's called distance-based method, I have seen this. Some people use the hyperbolic space before. So some actually there is some analogous to do that for the phylogenetic tree reconstruction. But I mean, maybe we might be able to do that, but I don't think that there is a good mapping between the tree space and hyperbolic space.
Okay, great. So, well, thank you for that very good talk. It's almost the end of the events in LOGML for today, but there is a social happening immediately after this. So we encourage everyone who can currently hear me to go. And details for it are on Slack, and you can see what joys are available. There's, for example, an escape room, there's code names, there's lots of stuff. If you check the games channel, what's it called, games-social channel, that contains a lot of stuff. So thank you, Ruriko, once again. And I will see you all possibly in 5 minutes or possibly tomorrow.
Yeah, thank you so much. Probably tomorrow, but thank you so much for your attention, and it was nice to meet you guys. So see you later. Bye.
Bye.